Okay, paper 2 short answer questions. So question 2, dihybrid crosses with fruit flies. Morgan discovered his results did not correspond with the expected Mendelian ratios. I explain this by suggesting that there is an exchange of genetic material between chromosomes, and this image shows his diagram for 3 gene loci on a pair of homologous chromosomes during meiosis. So what he's drawn is crossing over, and the stage of meiosis where that exchange of genetic material occurs is prophase 1. Now this is oft debated because some magazines will say prophase 1, some magazines will say metaphase 1. I always believe it's prophase 1, so I think let's always go for prophase 1. Explain the reason that Morgan's results did not agree with the expected Mendelian ratios in a dihybrid cross. So the expected ratios always assume random orientation and independent assortment. The genes are linked, so they show different ratios. And because crossing over had occurred, you would see some recombinants, but not the same as if there had been independent assortment. Okay, question 3. So we've got a membrane potential graph here showing membrane potential of an axon during action potential. So state the approximate value of the membrane potential at X. Okay, so X is nicely situated between the bottom and the top. Looking at the approximate values, X is going to be 0, isn't it? Because this is going to be 30. That's going to be minus 70. So that's going to be your 0 there. Okay. Y is the threshold potential. So state what happens when the threshold potential is reached. So at the threshold potential, you've got your sodium channel start to open, depolarization occurs, and action potential begins. It's that all or nothing response. Once it starts, it goes. Part C, describe the movement in ions that occur during time t. So that's from here to here. So that's the action potential, so the depolarization and then the repolarization. So you're going to have sodium ions are going to diffuse into the neuron in the first part, and then potassium ions are going to diffuse out of the neuron in the second part. Okay, those are your key points there. So then explain how a nerve impulse is passed on to other neurons. So it's going to be passed through other neurons through a synapse. Okay, so impulses are passed through another neuron at a synapse. So your depolarization causes the calcium ions to diffuse into the presynaptic neuron or knob. That causes the neurotransmitter to be released, and that then crosses the synaptic gap. And then the neurotransmitter binds to the receptors on the postsynaptic membrane, which then, as long as the threshold potential is reached, will cause the sodium gates to open in the postsynaptic membrane. Okay, so here we've got a nice picture of hemoglobin, conjugated protein consisting of 4 polypeptide chains with the attached heme groups. And so the first question is state the level of protein structure which the polypeptide chains of hemoglobin are combined. So that's many chains combining together, so that's called ternary structure. Question B was the one which asked you to explain the shape at X, but there was no X, so we will gloss over that one. So then part C, outline the role of tRNA in hemoglobin synthesis. Well, let's rephrase that question. Outline the role of tRNA in protein synthesis, because hemoglobin is a protein. So we're going to say it's involved in translation. It carries an amino acid that's specific to its anticodon, which then pairs with the complementary codon on the mRNA. It's only 2 marks here, so you don't need to write masses about translation. But just to outline that. Okay. And question 5, distinguish between the transfers of energy in inorganic nutrients in ecosystems. So remember that energy is it flows through an ecosystem, whereas nutrients cycle. So those are your key points. So the energy is going to flow through the ecosystem, being lost at each trophic level, whereas nutrients are constantly recycled and reused. So then part B, outline the role of methanogenic archaeans in the movement of carbon in ecosystems. So that's methanogenic, so that's going to be involved in methane. So it's produced, so methane is produced from organic matter in anaerobic conditions. It's then diffuses into the atmosphere, or it accumulates in the ground and it's oxidized to convert to carbon dioxide. That's more than 2 marks worth there, so you've got a little bit of flexibility there. And then describe how autotrophs absorb light energy for 3 marks. So you're going to say that it's absorbed by the photosynthetic pigments, that chlorophyll absorbs blue and red, or you could even draw the absorption spectrum if you wanted to. And you say that photosystems are a group of pigment molecules. They're located in the thylakoid membranes, and it causes the electrons to be excited to a higher energy level. So again, that's only 3 marks for something which is probably a little bit more complex than that. So you can just pick what you put in there. Okay, so then here we've got a tripeptide. So label 1 peptide bond in this molecule. So you could have had this one or this one. Okay, so those are your locations of your 2 possible peptide bonds, but you only labeled 1. Then describe the secondary structure of proteins. So that's the first level of folding. It can be either the alpha helix or the beta pleated sheets, stabilized by hydrogen bonds. So those are your key points. And then a kind of a random extra bit, outline the action taken by the diaphragm during inhalation. That's kind of come out of nowhere, hasn't it? So the action taken by the diaphragm during inhalation is that it flattens, it contracts and it flattens, and that increases the volume of the thorax. Okay, and that's the end of that. For section B, for the longer answer questions, please just consult the mark scheme. I'm not going to talk through the long answer questions there. All right.